Every instance of growth, healing, and reproduction in a living organism comes down to a single mechanical action, a cell physically dividing in two. But splitting a cell isn't like cutting an object in half. Inside the nucleus is a massive, highly complex database of DNA and organelles. The cell has to duplicate all of that hardware and genetic code without a single transcription error or physical entanglement. To solve this engineering problem, eukaryotic cells rely on a highly regulated, sequence-driven engine known as the cell cycle. Understanding how biological life sustains itself requires looking at the specific machinery of this cycle. A single replication error at this level can lead to the dysfunction or death of the entire organism. This diagram shows the complete roadmap of the cell cycle, an asymmetrical loop divided into two macrophases. The vast majority of a cell's life is spent in interphase, these large lower wedges, where molecular heavy lifting happens. Then, taking up a fraction of the timeline is the division phase, either mitosis or meiosis. This is the rapid execution phase where the cell physically splits. Long periods of invisible molecular preparation, followed by a sudden physical division. That is the operational rhythm of all biological growth. Interphase is often casually called a resting stage because the cell isn't actively splitting. In reality, it is the most resource-intensive manufacturing period of a cell's life. The manufacturing starts in the G1 phase. The cell rapidly increases in size while synthesizing essential RNA, ribosomes, and enzymes. Not all cells proceed. Some take an off-ramp into the G0 phase, permanently halting division to perform specialized jobs, like a neuron. If a cell continues, it enters the S phase, duplicating its entire genetic code. The original thread-like chromatin and its new copy physically link at a central node called a centromere. The result is identical sister chromatids, forming a distinct X shape. Finally, in the gap2 or G2 phase, the cell builds the physical machinery it will use to pull those chromatids apart, specifically replicating centrioles and assembling a framework of microtubule spindle fibers. By the time interphase is over, the cell possesses exactly twice the genetic software and internal hardware required to survive the mechanical stress of division. With preparation complete, the cell enters mitosis. This is the process of somatic division, designed to produce two identical daughter cells while maintaining a strict diploid or 2N chromosome count. Karyokinesis, the division of the nucleus, unfolds in 4 sequential stages, remembered by the mnemonic PMAT: prophase, metaphase, anaphase, and telophase. This diagram illustrates prophase. Loose chromatin condenses into visible chromosomes, the nuclear envelope breaks down, and centrioles deploy the mitotic spindle. During metaphase, those spindle fibers attach to the centromeres, forcing the chromosomes into a straight line across the cell's equator, forming a metaphase plate. Then, in anaphase, the central connection snaps. The spindle fibers reel the now separated sister chromatids apart, dragging them toward opposite poles of the cell. Finally, in telophase, the spindle fibers dismantle. New protective nuclear envelopes rebuild around the separated genetic data on each side. Because this sequence of align, split, and pull is so strictly enforced, it guarantees that neither side of the cell is missing a single line of genetic instruction. Now that the nuclei are separated, the final mechanical step is cytokinesis, the physical division of the cell's main body and cytoplasm. This illustrates animal cell division. The outer membrane pinches inward, creating a cleavage furrow that deepens until the cell snaps into 2 distinct daughter cells. Plant cells have rigid walls that cannot pinch. Instead, Golgi vesicles fuse to build a cell plate down the middle. Karyokinesis divides the genetic software. Cytokinesis cleanly divides the physical hardware. Mitosis builds identical clones, but sexual reproduction requires a different engine, meiosis. The goal here is a reductional division, taking a diploid cell and cutting its chromosome count in half to create haploid gametes. This diagram shows the mechanical divergence in prophase 1. Homologous chromosomes pair up, synapsing to form tight clusters called tetrads. This close proximity allows crossing over. Non-sister chromatids zip together at chiasmata and physically swap segments of DNA, creating entirely new genetic recombinations. This leads to a different geometry in anaphase 1. Spindle fibers pull whole homologous chromosomes apart, but the sister chromatids remain attached. This action effectively cuts the overall chromosome count in half. From there, the cell undergoes a second round of division, meiosis II, which mirrors mitosis to pull those remaining chromatids apart. The final result is 4 genetically unique haploid cells. Mitosis is engineered for exact fidelity. Meiosis is engineered for diversity, ensuring an entire species can adapt to environmental changes. These microscopic mechanics scale up to dictate the macroscopic results we see in the physical world. Because mitotic cloning preserves the exact integrity of the parent organism, it drives tissue repair, physical growth, and even asexual reproduction, as seen here in a hydra budding a new clone of itself. Meiosis, on the other hand, constantly shuffles the deck. By forcing genetic variation, it provides the raw material necessary for evolutionary adaptability. The discrete mechanical phases executing inside a single nucleus ultimately dictate the survival and shape of every living organism on Earth. But a biological engine this powerful has to be strictly bracketed. When cells bypass their regulatory checkpoints and divide uncontrollably, the result is tumor formation. To prevent runaway growth and shape normal development, the cell cycle includes programmed end states like apoptosis. This is controlled cell death, a clean, non-inflammatory removal of unwanted cells, like the targeted loss of a tadpole's tail. This controlled recycling is the exact opposite of necrosis. Necrosis is unplanned, toxic tissue death caused by physical injury, chemical toxins, or a lack of blood supply, as seen in the dead spots on this leaf. Mechanically, necrotic cells swell and violently rupture. They spill their intracellular materials into the surrounding environment, triggering harmful inflammation. The eukaryotic cell cycle functions as a highly regulated system, a tightrope walk between exact duplication, necessary genetic variation, and programmed destruction.